everybody. Hope you all are fine and are enjoying a good state of health. Today we are going to start a series of lectures about the general anatomical description of the nervous system. So at the end of this first session, the participant should be able to define nervous system, explain nervous system versus computer, classify nervous system, give a brief review of central nervous system, give, give a brief review of the early development of the central nervous system, describe the general anatomical features of the spinal cord, describe the location and coverings of the spinal cord, describe the external and internal features of the spinal cord, and lastly explain the blood supply of the spinal cord. Nervous system is an extensive, intricate network of different cellular structures which can perceive or recognize stimuli, transmit these stimuli, and activate certain cellular structures inside the nervous system. They and coordinate these stimuli, compare and contrast with previous experiences, and by making decision controls various functions of the body to maintain homeostasis. And this is achieved in collaboration with the endocrine system. Example is, as we can see in the diagram, adjustment of hot water tap. Nervous system versus computer. Nervous system has got a sensory system which is represented in computer by input devices such as keyboard. Nervous system has got central nervous system as the central processing unit. This is represented in computer as CPU, the central processing unit. Nervous system has got a motor system which is represented in the computer by output devices such as monitor. Now let us see what are the differences. Humans rely on energy derived from food, whereas computers rely on electricity. Humans are good at reasoning and creating, whereas computers are good at computing and logics. Humans are good learner, but the computer are better at multitasking. As regard the classification of the nervous system, first the anatomical classification. Nervous system consists of two parts, central nervous system and peripheral nervous system. The central nervous system comprises brain and spinal cord. Brain is responsible for processing of information received, initiate responses, stores as memories, and generate thoughts and emotions. The second part, spinal cord, conducts signals to and from the brain and also controls the reflex activities. The second part, peripheral nervous system, has two portions, sensory system and motor system. Sensory system consists of peripheral nerves, plexuses and ganglia. They convey information from external and internal environment from the receptors to the central nervous system. The second part of the peripheral nervous system is the motor system, which consists of peripheral nerves and plexuses and conveys orders to the effectors. that is muscles and glands. Second classification is physiological classification. In this classification, again, nervous system is divided into two parts, somatic nervous system and autonomic nervous system. Somatic nervous system controls voluntary movements, whereas autonomic nervous system is responsible for the control of involuntary responses. Autonomic nervous system is again divided into two parts or divisions. Sympathetic division, which is responsible for fight or flight, and the parasympathetic division, which is responsible for rest or digest, and also for the purpose of procreation. Now let us have a look at the central nervous system. It is the central processing unit of the nervous system and comprises brain and spinal cord. It is located in the cranial cavity and vertebral canal. It is covered by meninges, that is dura, arachnoid and pia mater, with cerebrospinal fluid in the subarachnoid space. It develops from neural tube and has got two components, wall and the cavity. Wall consists of neurons, neuroglia, and vessels, and is divided into two parts, the gray matter and white matter. Gray matter mainly consists of cell bodies of the neurons and the neuroglia, whereas white matter consists of processes of the neurons, which are also named as fibers, and the neuroglia. Second part is the cavity, which is of variable depth and have cerebrospinal fluid inside it. In the brain, there is a network of having variable depth of a series of cavities which are named as ventricles. And in the spinal cord, it is present as an elongated canal, the central canal. Development of central nervous system. First, the gastrulation. This is an early developmental process in which embryo transforms from a one-dimensional epiblast layer, a blastula, to a multi-layered and multi-dimensional structure, the gastrula. In this process, the epiblast cells at the periphery of the blastula proliferate and migrate towards the center. They invaginate through the primitive streak, and the first generation of these invaginated cells settle as the endoderm. The second generation insinuate between the epiblast and newly formed endoderm as the mesoderm. The remaining cells of the epiblast layer transform themselves as the ectoderm, which then forms the central nervous system through the process of neurulation. This is a process which is induced by underlying notochord, and the overlying ectoderm transforms as neuroectoderm in the shape of a neural plate, which later folds to form the neural tube. The cephalic two-third develops more and ultimately forms the brain, whereas the caudal one-third develops less and ultimately forms the spinal cord. Spinal cord is a thin, elongated tubular caudal part of cerebrospinal axis. It is located in upper two-thirds of the vertebral canal and is about 45 centimeter long and 30 gram in weight. It continues above with medulla oblongata at the level of foramen magnum and narrows below to a sharp end which lies at the level of disc between lumbar first and second vertebrae and is named as conus medullaris. There are 31 pairs of spinal nerves which are attached to the spinal cord. Each of these nerves is attached through a sensory root and a motor root. Spinal cord functions as number one, connection. It provides connection between brain and spinal cord for the stimuli. And number two, it functions as a simple reflex centers. As regards the coverings of the spinal cord, the spinal cord is protected from outside in by number one, vertebral canal, number two, meninges, that is dura mater, arachnoid mater, and pia mater. And it is cushioned by cerebrospinal fluid present both in subarachnoid space and central canal. Spinal cord is held in position by two factors. Number one, denticulate ligaments, which are 21 pairs in the shape of triangular shaped extensions of spinal pia mater. They extend sideways from the spinal cord. And number second is phylum terminal, which will be discussed in the coming slides. Notable point in this diagram is the presence of dura mater, arachnoid mater, and pia mater and their relationship among themselves. Now we start with the external features of the spinal cord. Spinal cord is nearly a cylindrical structure with relatively flattened anteroposteriorly. Its girth increases in cervical and lumbosacral regions, which are named as enlargements. These enlargements give large nerves for innervation of the limbs. Cervical enlargement extends from fifth cervical to first thoracic segment, and its maximum circumference is 35 millimeter. Second is lumbar enlargement, which extends from lumbar second to third sacral segment, and its circumference is 33 millimeter at its maximum girth. Now we come on to the fissures and sulci. These are longitudinal depressions of variable depth which extend almost throughout the entire length of the spinal cord. These are number one, anterior median fissure, which is about 3 millimeter deep on the anterior aspect of the spinal cord and contains connective tissue and blood vessels. Second is posterior median sulcus and septum. This sulcus is much shallow and from its depth a septum extends anteriorly, which is named as posterior median septum, and it roughly goes inside 4 to 6 millimeter deep. Third is posterolateral sulcus, which lies on the dorsal aspect and posterior rootlets are attached at this site. Fourth is posterior intermediate sulcus, which divides the dorsal white column into medial and lateral parts. Conus medullaris is the lower part of the spinal cord where it narrows and becomes conical with a sharp lower end, which lies at the level of disc between lumbar first and second vertebrae. Phylum terminale is a fibrous band made up of meninges. It is about 20 centimeters long and extends from conus medullaris to the coccyx. It is divisible into two portions. Proximal 15 centimeter, which is covered by tubular extension of the arachnoid matter, is named as phylum terminale internum, and the distal 5 centimeter, which pierces the tubular arachnoid matter and gets attached to the dorsal aspect of first coccygeal vertebra, is named as phylum terminale externum. Chorda equina is the horse-tail appearance in the lower part of the subarachnoid space. It is made up of conus medullaris, number 2, bundles of rootlets of spinal nerves, and number 3, phylum terminale internum. Why this appearance is present? Reason being relative shortening of spinal cord as compared to vertebral column, but the formation of the spinal nerves is fixed in the intervertebral foramen. Hence, due to shortening of the relative shortening of the spinal cord, these rootlets elongates to their formation of the spinal nerves. There are 31 pairs of spinal nerves which are attached to the spinal cord. Out of these, there are 8 in the cervical region, 12 in thoracic region, 5 in lumbar region, 5 in sacral region, and 1 in coccygeal region. Each spinal nerve is attached through two roots. First one, the ventral or motor spinal root, which is attached at the anterolateral aspect of the spinal cord. Second one is the dorsal or sensory spinal root, which bears an ovoid swelling, the dorsal root ganglion, which, which houses the cell bodies of the sensory neurons and is attached at the posterolateral sulcus. Spinal segment is the part of the spinal cord which gives attachment to one pair of the spinal nerves, or it is the part of the spinal cord which houses cell bodies of one pairs of spinal nerves. Internally, the spinal cord consists of gray and white matter. Gray matter is present in the shape of a fluted column, which is roughly H-shaped on transverse section, and gray matter itself comprises complex intermingling of predominantly nerve cell bodies, hence it appears gray, with their short processes, dendroids, neuroglia, and blood vessels. The white matter, which comprises well-organized neuron fibers, that is axons, neuroglia, and sparingly blood vessels, is segregated as anterior, posterior, and lateral columns or funiculi due to the presence of H-shaped gray matter. We first take up the gray matter of the spinal cord, which is present as symmetrical right and left comma-shaped masses which are connected by a transverse bar of gray matter, the gray commissure. This whole gives a shape of a letter H with concavity on lateral aspects. Anterior to gray commissure is a band of nerve fibers which crosses the midline. This is named as white commissure. These two commissures, that is gray and white commissures, are the only neuronal continuity between two halves of the spinal cord. Anterior and posterior limbs of gray matter, which are also named as ventral and dorsal gray horns. The ventral gray horn is present as a short and broad mass and is directed ventrolaterally. It does not reach the surface and remains separated by lateral part of the anterior white column. Second is the dorsal gray horn, which is somewhat narrow and is directed dorsolaterally. It is capped by a V-shaped translucent gelatinous mass of neuroglia having a few cells and fibers. This is named as substantia gelatinosa of Rolando, which correspond to the lamina 2 of the gray matter. It almost reaches the surface, but a thin lamina of white matter. The dorsolateral tract is interposed in between. Lateral gray horn of the spinal cord is a small spike-like projection on lateral aspect of gray matter in the thoracolumbar region. It extends from thoracic 1 segments to the lumbar 2nd segment of the spinal cord. It contains preganglionic sympathetic neurons which give preganglionic sympathetic fibers. They travel in ventral root of the spinal nerve and pass out as white rami communicant, which are 14 pairs in total. These fibers reach sympathetic ganglia of the spinal chain. Similar projection, not well demarcated, is present in the sacral region from sacral 2nd to sacral 4th segments of the spinal cord. It contains preganglionic parasympathetic neurons, which give preganglionic or pelvic parasympathetic fibers. Now let us have a look at the gray matter of the spinal cord at different levels. First, the cervical level. Both the posterior and anterior horn are bulbous and the lateral horn is absent. At basic level, both the anterior and posterior horns are slender and the lateral horn is present. In the lumbar area, the anterior horn is broad and thick, whereas the posterior appears slender, and the lateral horn extends down to lumbar 2nd segment. In the sacral area, both the posterior and anterior horn are bulbous and thick, and the lateral horn is absent. The H-shaped gray matter of the spinal cord divides the surrounding white matter into posterior, lateral, and anterior or anterolateral columns. Fiber in these columns are segregated as tracts. There are three types of tracts: sensory or ascending tracts such as spinothalamic tract, and motor or descending tracts such as corticospinal tract. These tracts connect the brain and the spinal cord in both directions. Third category is intersegmental tracts, which connect the various segments or various portions of the spinal cord within. The central canal of the spinal cord is the cavity of the spinal cord. It is extremely narrow tunnel which traverses gray commissure longitudinally. It is lined by ependyma, the columnar ciliated epithelial cells, and is encircled by a translucent zone, the substantia gelatinosa centralis, which mainly consists of neuroglia and a few nerve cells and fibers. This substantia is traversed by processes from the deep ends of the ependymal cells. The central canal continues above with the lower end of the fourth ventricle and ends below in conus medullaris as a slight dilatation the terminal ventricle. The central canal may extend 5 to 6 millimeter into upper part of phylum terminale. Now we come on to the blood supply of the spinal cord. First, the arteries of the spinal cord. There are two main groups. Number one are the longitudinal arteries and number two are smaller arteries. Among the longitudinal arteries, there is one anterior and two posterior spinal arteries. The anterior spinal artery is formed from the branches of vertebral arteries and travel in the anterior median fissure and supplies anterior one-third to half of the spinal cord. Posterior spinal arteries, which are two in number, each of them originate from the vertebral or posterior inferior cerebellar arteries and travel along the dorsal roots. Each of them supplies about one-third of the respective side of the spinal cord. Second group are the smaller arteries, which are actually the segmental arteries, which are small vessels which enter vertebral canal with the nerve roots. Each of them divides into anterior and posterior radicular arteries, which travel along with the respective roots and anastomose with the respective main artery, that is anterior and posterior. The largest of these segmental artery is artery of Adam-Quiz, which arises from the inferior intercostal or upper lumbar arteries and supplies inferior two-thirds of the spinal cord. Veins of the spinal cord and vertebral venous plexuses. Usually there are three anterior and three posterior spinal veins. These are valveless veins and form an anastomosing network on the surface of the spinal cord. They drain into internal, that is anterior and posterior internal, and anterior and posterior external vertebral plexuses, which empty into systemic segmental veins. Internal vertebral plexus, in addition, also drains into dural venous sinuses superiorly. Comments and suggestions will be welcomed. Thank you for listening and watching.